like to oh yes, so the meeting recording. So yes, as I was saying that we're stuck. Welcome to our last session of 2020, and with that, I would like to start by mentioning our sponsors. First, the BC Bioinformatics program at UBC and SFU, and the Genome BC and Ecoscope at UBC, the Ecoscope program and initiative, the Canadian Bioinformatics workshops, the local company here of Appcelera, and Langara College, one of more recent new sponsors. And would also like to remind everybody that VanBUG is associated with ISCB and two sister organizations at Montreal and Toronto, MonBUG and TorBUG. So with that, I just want to remind everybody with our schedule today. So unfortunately, we do not have a student speaker this time. So we're going to start with, as I'm doing right now, our announcements and then our main talk. And then after that, we're going to open the floor to more informal or open-ended questions interacting with around the topic of the speaker today. So if you have any questions, please ask them in the chat throughout the talk. And me and the other moderators would look at the questions, and then we would interrupt the speaker to ask the questions. Or if you want to ask your question yourself, you can also do that. So either in the chat or there's a raised hand feature in Zoom. So with that, I would like to very excited to introduce Dr. Natalie. Dr. Natalie Prystajecky is an assistant professor at the Department of Pathology and Laboratory Medicine at UBC. She's received her PhD from UBC in Pathology and Laboratory Medicine in 2010. Shortly after, she joined the BCCDC Public Health Laboratory. And she's been doing exciting work there. She runs two labs, the Environmental Microbiology Lab and the Molecular and Microbial Genomics Lab. And at these two labs, she uses emerging technologies like genomics and genomic sequencing to improve the surveillance of outbreak and outbreak investigation, to assess an outbreak investigation. So today, we're very excited that she's going to be presenting us with her work on genomic sequencing and genomics in the context of the COVID-19 pandemic here in British Columbia. So with that, I will give the floor to Dr. Natalie. Please share your screen and go ahead. Oh, I think you're muted.
Okay. Thanks. Sorry. Yeah, thank you for the introduction. I'm really pleased to be here presenting today. And so, as I understand, there will be questions throughout, and I suspect that I won't get through all my slides. And so I wanted to start with my acknowledgments. And that this is an effort that lots and lots of people have been involved with. We started doing the sequencing as part of a CIHR research grant. And I guess Genome BC and CIHR research grant, and that then quickly changed into sort of a routine service. And so we sort of transitioned this from being research being done in collaboration with our colleagues at UBC, Richard Harrigan, Hope LaPointe, and John Tyson, and now over time has been transitioned to a routine clinical test that's actually an orderable test at the BCCDC. So it is part of our routine scope of work. And so I wanted to acknowledge all the people that we worked with, and I really want to apologize if I've missed anyone. So I will preface my comments, my talk with some disclaimers. And the first is that I'm doing I'm at a bioinformatics talk, and I am in no way a bioinformatician. I do very little, I do some data analysis, but not like you guys. So I want to preface it with that. And so I'm here to more discuss with you the public health perspective of why we do SARS-CoV-2 sequencing. I'll share with you some of the methods that we've been working on. largely around why we sequence the way we do. I'll tell you a little bit about some of the analysis, but then largely we'll talk about some of the outputs, like what do we use this data for, and how do we see this really changing the way we do public health response in the future. So I gave my first talk on SARS-CoV-2 on February 3rd, 2020. We had just started testing. I think we'd had our first positive case recently. And I did the screen capture for that talk of how many cases there were of SARS-CoV-2 in February, early February. And I remember watching these numbers climb and really looking at the number of cases versus what we knew about the original SARS. And there was a very unknown situation. We didn't know if we were going to see it in BC. I mean, it didn't take long for it to appear. And so I contrast that with where we are today. Like, who thought that we would get to, like, I just can't even imagine that it got to this. I don't think that when this all started, mid-January of 2020, that we anticipated that we would be reaching 68 million cases globally. Like, that's just beyond comprehension. And one of the things about this virus has made it so different than everything else is it's unprecedented spread, and the ways we've had to respond as a society and as public health laboratories and various different, vaccine companies, everyone having to respond. But one of the things that was really interesting is the early discovery and the early discussion of where this virus came from. I mean, we knew that SARS came from these civet cats. We know that MERS came from camels, and we're really trying to understand, where did SARS-CoV-2 come from? And of course, we were all very interested in those early days in the seafood market, where it appeared to be emerging from. There were a number of cases were clustered around it, and these people tended to it tended to be workers or people who frequented this market, and this market was both a live and dead animal market. And so it did have that possibility of disease emergence in that environment. One of the things we know about all of these is that there is that linkage to bats, and there was probably bats serving as an intermediate host, and we still don't know 100% of where SARS-CoV-2 really emerged from. But what happened very early in this outbreak compared to some of the other emergencies of these other coronaviruses is that the sequence data became available pretty quickly, and this helped us understand where it sat in the phylogeny and how it compared to other coronaviruses, how compared to SARS-CoV, and how it compared to the seasonal coronaviruses. And this helped us to sort of predict what might happen, because given that it sits quite far away from the seasonal coronaviruses, we knew that there was not going to be any, probably unlikely to be any cross protection from antibodies that exist against things like the HKU or some of the other other seasonal coronaviruses. Likewise, we knew that it was closer to bats than to any other species. And so this started to help us understand where it may have emerged from, but and really starting to contextualize this virus in the context of disease emergence and what could the animal reservoirs be. And what we've seen so far is that the animal reservoirs do not carry a huge importance in this outbreak. Of course, there are some instances where it is, but really this is largely a virus that transmits from person to person. But what this, this early availability of sequence data allowed international tracking of the virus emergence and how they're related to each other, and being able to understand flight patterns, how people are, how linkage between people may occur. And this was, again, a slide that I put up from NextStrain back in February. It's crazy. NextStrain has like hundreds of thousands of genomes now. And there was 52 available at the time. There's 52 that were available, and they were looking at how they clustered, and we saw that people were clustering. And what was very interesting is that people from there's these 3 strains from or 4 strains from China clustering with quite closely with 1 strain from the U.S. And they were seeing that there was a number of samples that had gained 2 mutations compared to the reference sequence, and that there was many cases that were clustering together, and that they crossed different geographic regions. So we were already seeing international transmission. The other thing that made having that sequence available early, I mean, it came out mid-January. We only learned about this virus, at the end of 2019, early 2020, to be honest. and the sequence data became available really quickly. And that, initially that really helped us because we needed to be able to develop a diagnostic assay really, really quickly to be able to respond. Because of course, commercial assays wouldn't be available in time. And by the early release of that sequence data, others, we were able to develop an assay, within days of, the sequence was released. The next day we ordered the primers and probes, a whole bunch of them. And then we were able to validate initially on synthetic DNA. And then once we had our first positive on real sample, to be able to have an assay that is continuing to be used today. And this is just sort of gives you a timeline of where things happened. We were the first public release of the genome was on January 10th. We started testing our first samples on January 23rd, 2020, and it was on February 2nd that we had our first positive sample. And so you can just sort of see how this accelerated. I mean, generally when we do assay development, we did an assay development in 2 weeks to having it fully in place. Normally that took for us is a 2 to 3 month process, and just speaks to the accelerated pace. And so, none of this could have been possible without that early release of that genome sequence. And that has been part of the ability of public health agencies around the world to respond. So part of this is that, we moved from really relying on that sequence data to be able to develop the diagnostic tests, to be able to then move on to using that as part of our pandemic response. And so what we really needed to do is think about the strategies in which we wanted to do sequencing. And this is an approach that we would take for any new sequencing problem, is we have to think about the different ways in which we can generate the sequence data. So we historically have really worked on bacteria. Sequencing bacteria is pretty easy. You grow them, you have a pure colony, so you have no interfering information, no background, no host cells, no competing bacteria or other nucleic acid, and you basically do shotgun sequencing. You use some standard protocols, and it's the sequencing aspect is fairly straightforward. Of course, when you're working with a virus, it's a lot more difficult because we are not culturing the virus. This virus is not great to culture, and as a very transmissible virus, we want to keep the culturing to a minimum. And so we basically would have had 3 strategies to take. The first, and this is what was actually done early on in the pandemic, was to perform metagenomics. Grab a human respiratory sample, and sequence everything that's there. This, of course, has its limitations. The first being that you have to sequence quite deep. The viral material is just a small minority of the nucleic acids in a respiratory sample. There's going to be other viruses, other bacteria, and then a ton of human material, which has a much higher genomic density than what a little itty bitty virus would have. So from that perspective, you have to sequence extremely deeply to actually get enough viral sequence to be able to do an assembly. It is, however, the least bias in that you don't need to have any a priori knowledge of what you're sequencing, and you're not introducing bias by using primers or probes, which may select for certain types of viruses, if for example, if there's a mixture or if it's just not present. The next option is tiled amplicons, and this is, you have an a priori knowledge of the genome sequence. You generate across the entire genome these amplicons that tile across the entire genome, and you're able to sequence for a fairly inexpensive cost, and it's fairly effective and efficient. Of course, it's not without its own limitations. When you are tiling across a large genome, or any genome, I guess this is a small genome compared to a bacteria, but when you're tiling across 29,000 bases, that's a lot of primer sets, and there can be some primer kinetics that results in amplicon dropouts. So then you're always sequencing to the lowest common denominator. You also have challenges that by sequencing across the genome with all these primers, you have your primers perhaps masking some of the variants that are there. There's ways to deal with that. And then, of course, this is biased. And so if there are mutations that are occurring in the areas where these primers are binding, then you may not be able to sequence that part of the genome. And lastly, we have probe enrichment. And this is a completely different strategy. And I personally really love probe enrichment. We do it for a number of our projects. It's really great when you have difficult matrices or very little genomic material. And that's really where you start with, you make a library from a difficult sample, and then you use probes that again tile across the entire genome, and you enrich for your sample of interest. So that it's kind of like doing metagenomics, but you're only sequencing the parts that you really care about. And the nice thing about probes is that you don't have to have perfect matches, in that you can have up to, you can have up to 10%, if not more, of mismatch and still be able to pull down that genomic segment. And so from that perspective, it has less bias than the amplicons, but it's a very expensive technique. It adds about $100 per sample to your library prep, and it's time consuming, and it really depends on the quality of your probe design. So, here we went for the tiled amplicon approach because that's what the whole world is doing, but also because it had so many benefits of, in terms of the cost, that there were established protocols out there, and so from there we had to think about what primer scheme do we want to use. At the time that we started sequencing, so we generated our first sequence in February. The main protocol that was out there was developed by Nick Lohman and Joshua Quick, and they had used a similar strategy with primal scheme to sequence Zika and Ebola, and tiling the amplicons in a very specific strategy across the genome. And so that's what we started off with. And so the early the version that's currently being used by everyone is the third version, but generally what it is 98 primer pairs that are tiled across the entire genome. We separate out those primer pairs into 2 different pools, and we're generating 400 base pair amplicons with 200 base pair overlaps. Now, because there's so many primers, there's a possibility of a lot of primer interactions, and so that's why we separate them into these 2 pools. And in general, there are some primer pairs or some amplicons that just don't work as well as others, based on the sequence of the genome itself. And to overcome this, the strategy is just you sequence deeper. So you are trying to hit, if you're trying to hit 50 or 100 times coverage, and it's a poorly performing amplicon, you're sequencing extra deep across all the samples just to get the minimum coverage you're seeking for that area. And so this is just a visual of how the different iterations of the Arctic protocol have improved, and you can see that in V1 there were entire dropouts, like there were amplicons that just never worked. And then you can see that as the primer pools, the sequences were adjusted, and then also the ratios were adjusted. You can see that you can improve the yield in each of those primer pools and be able to, for example, in this instance, be able to at least hit 50 times coverage for all of them. But you can still see that there's variable performance in all these pools, and some, in some instances you'll get 50 times coverage, like this last one here, and other times you're getting well over 1,000 times coverage. And so that balancing of the yield on the amplicons is something that was still problematic for us, even after we'd been running this protocol for months. And so, there a number of different primer schemes came out after that, that were based off of the Arctic protocol, so using the exact same primer sets, but actually just pulling out some of the primer sets to generate bigger amplicons. So the original one had 400 base pair amplicon size. You can sort of just see that you're getting pretty sort of this jagged performance over the course of the genome. And it's not bad when you have a sample with fairly good viral load, but when you're going on to a sample that has less virus in it, so this is what you can see here, it says CT20 and CT31, so this is a sample that's strongly positive, this is a sample that's more weakly positive. You can see that the dropouts happen pretty frequently compared to when you have a higher viral load. And for us, we're often asked, can you help us find patient 0 of this outbreak? And if patient 0 only has a sample with a higher viral load, then we have to try and sequence it. So this is a paper, sorry I didn't mention the author's name, it's Nikki Freed, and she's from New Zealand. And this is a paper that explored pulling out some of the primer pairs to create amplicons of different sizes. And so they tried 1,200 base pairs, 1,500 base pairs, and 2,000 base pairs. And what you can sort of see is that, you know, you're not at the lower viral load sample, this sample with a CT31.2, you are still seeing some drops in coverage, but you never see complete dropouts. And, you know, part of this is that it's probably an improved primer interaction, so that you are able to so that the amplicons are amplifying more consistently. So we're getting more reliable coverage when we moved on to the 1,200 base pair protocol. But the one challenge is that if there's a mutation in that primer site, you know, you'll end up having a 1,200 base pair dropout, as opposed to with the Arctic V3, where you'd have only a 400 base pair dropout. So it's really balancing, you know, whether or not you know what's more important. And so we did lots of validation to compare the two, and we ultimately decided to move on to the 1,200 base pair protocol. And this is just a diagram showing coverage plots across the genome for two different samples. This one here, this is one set here, and this is another set here. And with the 400 base pair protocol, like, I mean, the coverage is pretty good overall. And if you were to average out that coverage, I mean, we're probably, you know, we had probably, you know, I don't know for the sample, 80, 90%. But what you're seeing is these like tons of little dropouts happening. And so you're getting regions of the genome where you're not able to get as consistent sequence quality. And so when we move to the 1,200 base pair protocol, we see more consistent sequence data, less of this noise. And in fact, in this sample, we have a beautiful sequence there with no dropouts at all. And both of these were difficult samples. And, you know, that's really what we're trying to work towards, so that more, you know, for samples that have a high viral load, both of them work really well. And in fact, the 400 base pair one works a little bit better for the good samples, but it's those, you know, those edge cases where we really do want to be able to generate that data for, that we found that this, the 1,200 base pair protocol worked a bit better.
So I have a question here.
Yeah.
So does it make sense to generate like a mix primers that are of different lengths, of different sizes? And can that like help you with like the issue with like when you have like mutations in the sample, but you want like have like the better coverage overall, but like, just in case you have mutations? Yeah, so you know, I think it depends on your sequencing, your strategy for creating your library. So you can, you know, if you are using if you're using enzymatic tegmentation to be able to generate libraries, if your fragments are all sorts of different sizes, you may overfragment the smaller amplicons. And so then when you're doing your bead-based cleanups, you're not sort of dealing with this, you know, everything being a similar situation, like the similar size. So from a wet lab perspective, I don't think that's ideal. But it's certainly, I think if you were to, yeah, I think it's not it's generally you want to have sort of consistent insert sizes. Yeah. I see. All right, so yeah, so we moved on to using the 1,200 base pair protocol in Illumina. Now, when we started this project, we actually started doing it on the MinION. And the Arctic protocol was originally designed for MinION, and all the analysis pipelines were originally designed for that. And there's a few reasons. I mean, the startup cost for MinION is negligible. And it can generate data pretty quickly. And it's relatively inexpensive. Now, the one thing, you know, the one reason we had to transition from MinION to Illumina is that we had an operational need of, you know, trying to be able to sequence up to 750 genomes a week. Right now with MinION, at the time that we started this project, we could only sequence 24 genomes at a time. That's how many barcodes were available. They do have 96 barcodes now, but the quality of data when you multiplex 96 samples on the MinION is not great. And so from that perspective, you know, we maybe could have multiplexed 48 at a time, but then if you imagine, if we're trying to do 750 genomes a week, we'd be just like eating flow cells, like it was no one's business. And just operationally, it's difficult to manage. And
the other thing is that you know, some of the protocols for the MinION weren't as compatible with automation. And so one of the nice things about some of the Illumina products, some of the Illumina library prep systems, so the NextShare XT and the DNA Flex, is that there are programs that have already been approved and pre-programmed onto a number of different liquid handlers. And so from that perspective, it was easy just to sort of plug and play to move a library prep onto a liquid handler, which is the only way we could to be able to reach this goal. And in fact, we haven't reached this goal of 750 right now. We're doing 450 a week. And then the last two things were that, you know, we were doing MinION largely as a research project, and it became clear that it needed to be a clinical service. And we have other sequencing programs as a clinical service. And so we do all the enteric bacteria, as well as some antimicrobial resistant organisms by Illumina sequencing already. And ideally, we would be able to just put, you know, Salmonella and E. coli and Klebsiella and COVID all in the same run. So we could just be churning out data that is disease agnostic or pathogen agnostic. And so that really wasn't possible with MinION because we weren't using MinION for those other purposes. And we already had a workforce that was already trained in Illumina sequencing. So that's really why we went that way. And you know, part of it is also just the consistency of data. And this is just an example of some difficult samples that we were having trouble sequencing on MinION. We were able to generate higher quality and more coverage on Illumina, which might just be, the protocol was done a little bit differently. We were always doing cleanup of our amplicons before sequencing, but it might just also be the Illumina technology. One thing I should point out is that even with our improvements, that we found that our approach using the 1200 base pair protocol on Illumina, that we were able to get sort of better performance than difficult samples on the 400 base pair protocol on MinION. We still see that we sometimes have difficult samples, and that beyond a CT30, so this is a viral load, you know, that's getting to the point where, you know, people may be shedding, but not actually infectious. We're not entirely certain. It also could be the beginning of infection. They're about to get, you know, quite a high viral load afterwards. But at a CT30, we see the sequencing quality drop off substantially. There's just not enough material there to generate a high quality data. That's not to say that you can never generate good data above CT30. You can see up here, you know, these are, you know, genomes, you have greater than 85% coverage, you know, up to a CT, you know, this looks like a CT36. So it's not impossible, but, you know, your sequencing yield for those high CT samples are just, or, you know, samples with low, not that much virus in them is just not great. It's not a good investment to do so.
We have a couple of questions from Faiza. So the first one is, what do you think causes the low coverage in MinION? And the other one is, what do you mean by difficult samples? Is it just related to the CT number or something else as well?
Yeah, so I think, I mean, with the MinION data, I think that I think we haven't done the 1200 base pair protocol on MinION. I think it's simply some of the primer interactions and that there are amplicons that are difficult to sequence. And so then you get these constant dropouts. So either there's mutations at those sites and they're just dropping out. And then you're, you know, based on where they selected to do the 1200 base pair protocol for the free protocol, you just sort of have more stable, more stability in that genomic segment. Or it could be just the primer interactions, and we know that we have to sequence quite deeply to overcome some of the challenges. And when you have a fragmented genome, it's just, you're seeing more of it. The difficult sample question, generally what we say is a difficult sample is a sample that either has not that much virus, so a high CT or a low viral load, or has been poorly, that has maybe not been maintained well, like not refrigerated, might come, you know, maybe a couple of weeks old, has not been taken care of. So, you know, from that perspective, that would be a low quality sample. Because
so would that reflect in the fragmentation of what you're seeing?
Yeah, so it would be, yeah, so a sample that's old could be, the virus could be over fragmented. A sample that is late in someone's infection, so we know that people can shed the virus in their nasopharynx for weeks after they're probably infectious. So you see, we have a population of patients that will shed the virus for 3 months. They're not infectious. The CT value, like the very, very little virus, and it's probably just remnant viral fragments that have no like impact on infection. And trying to sequence those aren't great quality samples. So a few different reasons, yeah.
Thanks.
So analysis. So this is, so we've, sequenced. We've generated sequence off the MySeq, and what do we do? What do we do next? So here at BCCDC, we follow the Connor pipeline, which is out of the COG UK. It's basically a Nextflow wrapper on, it's Nextflow on with a few additional features, taking the original Arctic pipeline and adding some features that are specific, in this instance for Illumina. For example, Connor has a fork where you can either generate the equivalent data, you can do the equivalent analysis off of MinION data or Illumina data. There's, most pipelines have generally a similar strategy. You start with quality, you start with quality filtering, make sure that you immediately would take out, sequences that were longer than you expect, because that's probably a chimeric sequence. Really short sequences can be problematic as well, representing overfragmentation. So that would be some of the quality filtering as well as adapter removal. Generally, you can map against the reference first. Some pipelines also will map against human as well, and then remove all human breeds before analysis. We're not doing that yet, but that we will be doing that soon. And then, the next step is really removing the primers and generating the consensus sequence. From there, once we've generated consensus sequence, we do a few different analyses. We would run it on a pipeline called ENCOVE Tools, developed by Jarrett Simpson at OICR. And we're using this really to understand, it's largely a QC tool for us to understand, samples with excess ambiguities, flagging samples that have high CT values. So it takes into account some metadata, and really ensuring that we're, really interrogating the data appropriately to identify high quality high quality consensus sequence for downstream analyses. We'll run Pangolin, so that's the more recent version of Pangolin, to assign a lineage. And that's also done in ENCOVE Tools, but we do it separately, just because sometimes the version in ENCOVE Tools isn't as updated as the one that we're using. For samples of interest, so like if we're doing an outbreak investigation, we'll right away create a SNP matrix just to see how the samples how they compare to each other. Are they the same or different? And through that approach, we're also able to generate mutation lists. And then what we're largely dependent on right now is we build a phylogeny where we integrate some basic epidemiological data on a local instance of NextStrain. So this is an example. This is the most recent up-to-date data up till last night of all the different lineages that are seen in BC. This data, we only have it for this version only has the Illumina data. So this is actually just data from mid-August to now, and you see we have some predominant lineages here, and we're able to sort of toggle between different views so we can be able to interrogate the data. Here is one where we're able to look by health authority and be able to see, what lineage or which sequence types are found in which health authorities. We're able to overlay whether or not we know these samples are outbreak associated or not. And so, and then we can then drill deeper. So this is an example of looking at whether they're outbreak associated or not. Here we're looking at the phylogeny of these samples based on divergence, so how many mutations they have versus the Wuhan reference strain. And we can also look at it in terms of a time tree and look to see something like, all of these are identical to each other. If we look back here, these ones are all identical to each other, but there is a time component to them, and understanding what that means in terms of who was involved in the outbreak and who was the first case to have been tested. Of course, there's a lot of biases when we bring in time tree, simply because we use the collection date as a indicator of time. We have access to data of onset, but that's not a very reliable data source. But then, you can see that there's sort of these pileups where everyone is on, from an outbreak is on the same date. Well, that's probably because there's been a mass testing event. And so does that really reflect anything about the transmission? So, I think that you have to, recognize that there's some biases in the data. But, being able to see who has been tested first or has the first date of onset can have some information, can help in the interpretation of the tree.
There is a question about earlier, like the sequencing. So it's about the dropouts. So I think it's from Kara, which is, what do you do when you see dropout from a mutation in a primer site? Do you resequence any of those samples with alternative primers?
We haven't. Generally we're sequencing from an outbreak, and so if there was an instance where the primer dropout happened across the entire outbreak, then we probably have to go back and do resequence it. But generally, what happens is you have a sample where you have a primer dropout, probably because of low quality sample, not mutations, and then there's other people in that outbreak who generated high quality data to help interpret the result. We have done some instances, we do have instances where, for an unexplained reason, we have poor performance on the 1200 base pair, and we have done the 400 base pair, and we're able to generate better data. So we've talked about having a rescue protocol for difficult samples where, it doesn't work the first time with the standard protocol. What strategy would we use for rescuing the sample? So an alternate sequencing scheme, perhaps we just we're just in the process of resequencing some samples that ought to have generated better data. Or do you use a different strategy like probe enrichment as well? So generally, right now we just sort of move on because we're often sequencing others in that outbreak, but there are times where people really care a lot, and we try to do what we can to improve this.
Mm-hmm. So how do you do the sample selection? I mean, I guess if you start from the universe of everybody who gets tests here in BC, how do you get from there to the people that test positive, the people that get sequenced in the end?
Yeah, so we had a pretty low caseload up until August, and up until August we sequenced everyone we could. So samples are tested in various labs across BC. They're under the Public Health Act. They're forwarded to us for surveillance, and we were sequencing any sample that came to us that had a decent viral load. And
so those ones that tested positive through a PCR test.
Yeah,
that exactly. That are positive.
Yeah, PCR positives only. And then all of a sudden things exploded, and we just can't keep up with that pace. And so what drives our sequencing selection is usually it's generally targeted. So right now there is a request process that a hospital or a medical health officer will submit a request for sequencing, and we will sequence samples related to an outbreak. And they will have very specific questions they're hoping to confirm, transmission networks that they're trying to use the genetic data to overlay to understand what's happening. Outside of that, if the runs aren't full, we were for a 3-month period sequencing all pediatric cases, and we were doing that as part of the support of understanding the reopening of schools. And then we also sequence healthcare workers, because there's always questions about transmission within healthcare workers. If there's extra room on a run, we just randomly pick from racks, and just a little bit of roulette, just pick random samples that represent the geographic diversity of samples received in BC. And so that sort of creates sort of that background. But I will admit, though, in the last month, there is no background being done, because it's all the sequencing is focused on outbreaks right now. And so we're hoping to build capacity in the upcoming weeks, post-Christmas, so that we can start to do more background sequencing.
Mm-hmm. And this is all from the same PCR swab that was done when the patient came in for testing, right?
Yeah, so it's all off the diagnostic swab. So that same, an aliquot of the viral transport medium or saline gargle is sent to us from those frontline labs, and we would re-extract and sequence.
I see. Okay, thanks.
Yeah. So why do we sequence in public health? So why are we generating all this data? Why are we why are we generating almost 450 samples, 450 genomes a week? And we're doing it largely for 4 reasons. And the first one is really what drives the majority of our work. We're doing it as part of outbreak response. There's confirmed epidemiological, we use it to confirm epidemiological relationships in disease transmission. The second is what we're hoping to get to, which is the cluster detection. We would like to be able to say, hey, all these samples are related to each other. You haven't asked us about them, but these are forming a unique clade. You should probably investigate this further. And in fact, this is the way it really works in foodborne disease investigations. We often see clusters of cases clustering together before we actually know that they're linked. So this is sort of driving, instead of it, we're sort of providing an epidemiological hypothesis as opposed to responding to an epidemiological question. We're also doing it to generate surveillance trends to understand the distribution of pathogens or trends across the province. And lastly, what we're also interested in is looking for genomic features. So right now, of course, there's many questions about the spike protein mutations and how this might impact immune response, particularly in light of the news about the vaccine being approved. What will this mean in terms of the diversity of virus that we see in the province and escape mutants, for example? So those are the main reasons we sequence, and I'm going to give you a few examples for these of how we've used this data. So for outbreak response, so this is an example. We were asked, there's these hospital clusters, there's a cluster in a hospital, they're on separate wards, and are they related to each other? Do we have transmission in the hospital that we didn't understand? And so what we can see here already is that what's sort of showing up in darker color are the cases. And in fact, you are seeing some clustering, but there's lots of sporadic cases as well. So yeah, there's a little cluster here and there's some sporadic cases here. And so what's happened is that we are seeing that we are able to distinguish who's related to one another in that hospital environment and confirm that, in fact, there is not transmission in the hospital. So so in this situation, you see these people who are, clustering quite nicely over here. And then, a number of cases who've acquired a mutation relative to the main cluster, but these are all outbreak cases and they're clustering together and it's a nice story. Here is an example where you have one outbreak case or one person who is from that hospital facility, and they are clustering not with the rest of the people in the hospital, but with a number of community people. And so it just suggests community acquisition and not transmission in the hospital. So, of course, these are non-outbreak cases, but they could be linked. And we had no evidence, and we were not asked, or we don't know anything about them that would link them. They, if you look at their epi data, they all say unknown exposure. So perhaps these cases are actually related to each other and should be looked at in further detail. So that really brings us to the second scenario, where we talk about, can we actually proactively say that these are genetic clusters and these are worthy of investigation and be able to communicate that to our EPIs? And so the one thing is, for us to be able to communicate to the EPIs, we need to decide a SNP cutoff to say these cases are related to each other and are worth looking at. And so do we say only cases that are identical to each other? Do we look at ones that are within one mutation of each other, one SNP from each other, or do we use more SNPs? And how do we define what that cutoff is for when we can say, hey, EPIs, you should probably look into these. So we actually did we did a manual investigation of looking at the concordance between genomic clades and the EPI outbreaks. And after this manual process, we are in the process of doing this again with a program called Tree Cluster with a few, we've made some few modifications to it to really serve our purposes and hope to be able to start to do this in a routine way. So this is just an example of the data set. We had 689 genomes from 609 COVID cases. We were then able to look at the clade definitions of how they look if we were only to use 0 mutations as our cutoff. So in this instance, we would say, oh, there's 3 different genetic groupings that are worth looking at. but we know that these are actually part of one big outbreak. We know they are all related to each other. So that definition of 0 mutations is probably just too tight. So if we go to, what if we went to 3 mutations? Well, if we went to 3 mutations, we do end up including everyone, but we start to include people who are not necessarily part of the outbreak. So you have to find that balance between what is the perfect cutoff to say, this is a cluster worth investigating. So we went to, with the data, we took over those 690 samples, they represented about 20 plus different, they represented 20 known epi clusters. And then we were able to do some stats to really look at sort of a sensitivity and specificity analysis. So sensitivity meaning how many of the epi cases would be captured if you used different genetic cutoffs. And then, of course, when we also looked at how many additional non-related cases do you bring into your tree when you increase the number of mutations. And we sort of landed on this sort of, from the manual inspection perspective, a sort of a sweet spot of how many variants are acceptable when generating this type of genetic clade-driven investigation. So the third scenario that we talked about is just looking at sort of population level surveillance and understanding what is happening at a larger scale than just the clusters themselves. So this is an example of the lineages changing over time. And what, if we were looking, the pangolin lineages they don't have that much resolution, but they do tell us, sort of the patterns that we're seeing. And so, what we're seeing is, there was, early in February, we sort of had a 50-50 mix between the B1s and the B11s. And over time, in March, when we had unfettered, access to our borders, and, we had that shutdown of our borders just before spring break in March, but still there was a lot of travel happening at that time. So between February and March, as our caseload increased, we started to see more diversity of virus. Then the shutdown happened, and we can see, again, continued shift in the variants. Now, June's an interesting month because there wasn't a lot of transmission. It was all basically like one lineage. I mean, there's other genetic subclades within it, but we see as things opened up in June that we see, again, diversification of the lineages that we see in BC and the emergence of a new dominant lineage, which is this B.1.1.113, which continues. If I didn't put up the November data, but you would see that it continues to grow. And that's a lineage that's largely associated with cases in Fraser Health. We are also, as I mentioned, this is an early diagram of sequencing all the pediatric cases in BC. And we're doing this to be able to support knowledge about how transmission is occurring in the school environment. And so you can see in color are the different age groups. You can see that there's no real, that that pediatric cases occur over the whole tree. Oh, I took out there. Sorry, I'm missing a slide there. But what we do see in this instance is that if we do look a little bit further into the clusters with the pediatric cases, we don't necessarily we see them clustering largely with community cases and not necessarily school-specific clusters. There's a handful of school-specific clusters, but the majority of the school-based spread appears to be related to community transmission. So lastly, I'd like just to bring up the topic of looking at
sorry, for that last slide. So do you also using genomic sequencing data that you get, do you have an idea about transmission not only between students but also between adults and students?
Yeah, so we do have so we were sequencing. There is we were sequencing adults that were associated with schools, as well as just general adults in the community. And what we were able to see, and sorry, I took out the slide for some reason, is we see these large clusters that form where there's a couple of pediatric cases, and there's a lot of adults in those clusters as well, and they're not necessarily adults that are associated with schools. And when we do see a couple pediatric cases in a single cluster, often there are sibling pairs, which suggests that there's transmission in the household as opposed to transmission in the school, because we don't see further transmission in that school environment. And that's really sort of what we've also been seeing in terms of the schools, just that often you see, we see a transmission event, or we see a notification that there's been a person who's positive, but not necessarily these onward large transmission events.
I see.
So lastly, I call it genomic features. You could call it mutation analysis. You know, often, before COVID, we would be looking for genomic features that were associated with pathogenicity. So looking at you know, which genes you might have that encode toxins in foodborne disease, or genes associated with antimicrobial resistance. But in the case of COVID, there's, you know, consistent, oh, there we go. My slides are a little bit mixed up. Here are some of the examples of adults and children clustering together, that there's very few pediatric cases in these contexts of these large genetic clusters. So here we go, back onto the mutation. So of course, people are interested to understand what changes in the viral sequence result in perhaps changes in viral fitness or infectivity or immune response. And so this is an example, I'm sure all of you have heard about the D614G mutation. You know, it was probably the first one described, and initially what they showed was that you know, in areas that had really severe illness, so for example in New York and in I believe it was Spain, that, you know, that the variant 614G was dominant as opposed to the wild type. And, you know, people were arguing for a long time that this could just be a founder effect. The fact is that that's the main strain that spread, not necessarily that had any fitness advantage. There is some lab-based literature now that has demonstrated that, you know, that virus with that mutation can replicate at a higher rate in cell culture. And so perhaps it does improve, result in higher replication, which means a higher viral load, which means more transmission. And so of course, you know, there's people, I don't think the case is closed on this one because people are still talking about it. In BC, at the beginning of the outbreak, so between February and August, we saw sort of a split. In the very beginning, we saw predominantly the wild type mutation, and then with time we saw, so here we see early on in the outbreak, we see a mix between the, you know, here is wild type and here's the mutation. And then with time, so by the time we get to August, we are not seeing we aren't seeing any of the wild type. We're only seeing the variant, the G. And if we look at our data since August, you know, our Illumina data, now we don't see any. And I suspect that these few little remnants that we have in our tree, there's a few cases in there that are real, and some of them are actually just reference sequences from before. So, you know, it's interesting to say, you know, it has this fitness advantage, and perhaps that's why it is the dominant strain. Perhaps it's just a founder effect, and that's all that's being spread right now. So we will be looking at outcomes data to understand during the earlier phase, when we do have a mix, whether or not there's any difference in the mortality outcomes or hospitalization outcomes for this within our province. There's another one, this is the N439K mutant. Again, this is a mutation in the spike protein, and it appears that this variant, so that organisms with this variant seem to be able to evade immune response. So they're resistant to neutralization with antibodies. And so in BC, we have a few cases. You know, they all had good outcomes. They did not, they seemed, you know, none of them were hospitalized. There was nothing about these cases that were particularly interesting. So, but that variant does occur here in BC. And so we're constantly asked, you know, do we have this variant? Do we have that variant? And, you know, our ability, this genomic sequence data really helps us understand that, because if there is a variant that's resistant to antibody response, that's going to be hugely problematic if it becomes a dominant strain in BC. And lastly, I just wanted to mention the mink. We heard a lot about mink in Denmark and in the Netherlands, and now, in fact, we have some interest here in BC. And then what has been observed is that there is transmission between human minks and back to humans. Mink have a respiratory system that's similar to humans, and they seem to, you know, generally there's a number of respiratory pathogens that mink are susceptible to. So they are a potential host. And so what they were worried about here is that there was, you know, zoonotic transmission from humans to mink and then back to humans. And, you know, from mink to humans, they accumulated a few additional mutations that may confer resistance to immune response. So of course, it was a huge problem in Denmark, and they resulted in depopulation of a lot of farms. So what they observed in that transition between mink and humans was a constellation of 5 mutations that were associated with mink, that they were very concerned about what the impact might be on human response to the immune system or vaccination. And these are the constellation of 5 mutations. They call it the cluster 5. And we've searched, we do not have any of them here in BC. So that's just some of the ways that we use that data. So what's next? You know, it's hard to think about what life post-pandemic might look like. But I think that, you know, particularly for trainees, it's really important to think about where you will fit in into this post-pandemic world, particularly if you're graduating soon. And, you know, I think that there's a precedent has been set. You know, I was on a call yesterday where I was explaining some phylogenetic trees to some medical health officers, and they're like, well, why don't you do this for flu? Like, when can you do this for flu? And when are you going to do it for this? And are you going to do it for RSV as well? And so I think that what I'm saying is that the precedent has been set, in that, you know, we were able to ramp up and start doing genomics on this pathogen pretty quickly. And we've been wanting for a very long time to be able to expand our genomics program at the BCCDC, but I think it'll become, you know, routine that we are going to be doing genomics for most pathogens in the future. I expect that we're going to be expanding our surveillance tools to a wide range of pathogens. Influenza, we've already been doing a little bit, but to expand that further, tuberculosis being the next one as well. And that it's going to go beyond just whole genome sequencing. And we're already seeing other ways in which genomics can be applied. And so, you know, how this virus was discovered was metagenomics. And we have lots of cases of encephalitis, for example, where we have no known pathogen. And so this is definitely the way to go. There's also, you know, Twist and Illumina and, you know, Swift Biosciences. They're all creating these enrichment panels so that you can start sequencing respiratory panels quite easily without having to have an amplicon scheme. So I think these are sort of really the ways of the future. And, you know, having trainees that have a really strong understanding of public health genomics, the wet lab, bioinformatic aspects will be really important sort of as our next round of improving the type of testing we do in a public health lab. And of course, you know, we will be, you know, we will be ready for the next one. I think this was certainly a learning lesson about the types of ways that labs need to be nimble to be able to respond to public health threats like this one. And with that, that's all I have to say today.
Thanks a lot. That was a great presentation. And I guess we have one question here from Angela. So thanks for the great presentation. And is it problematic to use epidemiologically linked outbreaks as gold standard in sensitivity and specificity analysis to determine the cutoff for the number of mutations? And how likely is it that epi-linkage is not indicative of true transmission association?
Yeah, I mean, we've dealt with that many times. And so one of the things is, like, we'll be asked to investigate an outbreak, and we'll get this like massive outlier who makes no sense. And then we'll go back to the health authority and say, like, you know, here are all these people they cluster together. It's a nice story, but then what's going on with this person? Like, oh, they just want to know where they got their pathogen from. Like, well, that's sort of problematic. And so, you know, what do you, like, I mean, if you use genetics alone, you know, it's going to be hard to create that cutoff. We could just say everything identical should be investigated, but we do need to be able to sort of link it back to the types of work that they are typically doing. And so we do look at sort of the high quality outbreaks, or the ones with really, you know, there's different levels of certainty when it comes to an epi investigation. And so, you know, there are certainly limitations of doing it that way, but largely the cutoff is supposed to be there to inform the epi. So we need to have that linkage back to that epi data.
Thanks. And we have another question from Andy. Have you tried any of the TE panels? I'm not sure what is that. You mentioned from some of the vendors.
Yeah, so we have done Twist's probe enrichment, and we have done Illumina probe enrichment for different projects. So the twist enrichment we tried on samples with a really high CT value, so a CT 35, which is sort of the equivalent of a single cell in a sample. Yeah, and it didn't work particularly well. It didn't work any better than amplicon sequencing. It was interesting for that data that the amplicon and the enrichment generated different data. So when we actually pooled it together, we got better coverage. But it wasn't great for the high CT samples. Now we gave, I feel like we didn't give the probe enrichment panel a fair chance because we like tried it out in the most difficult samples there is. So we will try it again. And then as a separate project with a collaborator at the University of Regina, we have used the Illumina panel to be able to sequence coronaviruses from humans as well as from animals. And it worked pretty well. The on-target rate wasn't bad. Yeah, I mean, we will be validating and exploring it further, and I think it sort of is the next big thing. But one of the things with the Illumina panel is that it was validated for diagnostics, not necessarily genomic characterization. So we'd have to really see whether or not how good the quality of data is coming out of those genomes compared to the amplicons. But it's beneficial in that it has a little bit less bias than the amplicon approach.
Well, I have another question a bit more general. Like, how do you compare BC's approach when it comes to like whole genome sequencing in relation to public health and pandemic in the context of COVID-19 to like other jurisdictions? Like, are other provinces in Canada doing similar work here? And is there like any aggregation of this data Canada-wide or maybe at like Canada, US, North America scale?
So I should have put in a slide about this. And there is a project called Cancogen, the Canadian COVID Genomics Network, I guess. And it is a project that received $40 million in funding from the federal government, $20 million for sequencing human samples to understand host response, and then $20 million for viral sequencing. And that's administered from Genome Canada. And it has academic and public health collaborators from across Canada. So there's a network of us. We meet usually a couple times a week, it feels like, where we discuss that we're doing the analysis and generating sequences in a fairly similar way. And we are transferring our data to the National Microbiology Lab in a centralized repository where they can start to do national surveillance. Whether or not we're doing things all in the same way, like on the wet lab side, what was sort of problematic is that Cancogen came to existence well after everyone across Canada was already doing sequencing. So there isn't a standardized protocol, but we are generally all using a different variation of the same thing. So we're using this 1200 base pair protocol. Alberta is using 2000. Ontario's using the 400 base pair protocol, but they're all sort of based off each other, so the data is very comparable. And then on the analysis pipelines, everyone is using, a variation of each other's, like it's all sort of forking off of, the Connor or the original Arctic. So it's sort of all being done in a similar way. And how we do things in BC in terms of the interpretation might be different. Because we have a really close relationship with our EPIs and we have access to the EPI data quite easily. Other jurisdictions don't necessarily have that capacity, and some of them are more focused on surveillance and less for outbreak. So it really depends on the province's priorities in terms of how they want to use that sequence capacity. But there is sequencing happening across all of Canada. Every province and territory is participating in the sequencing effort.
And like scale-wise, like is it similar to like, because I think you mentioned like here we're doing like 750 cases a week?
We want to do 750, but we've only gotten up to 450.
Okay.
And so for our caseload, we're generating a lot of data compared to some other jurisdictions, but then also, Quebec is generating more than we are because they have such a high caseload and high population. So, we're sequencing a good chunk of our samples. Lately it feels like a small chunk because the caseload's so high and we're only doing 450 a week, but relatively speaking, BC has a high proportion of data.
I see. Awesome. Thanks so much. So I think at this point we can stop the recording, and if people want to talk about this or have more questions to Natalie, please go ahead. So feel free to do that. And let me just ask